Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 140 us seminar, and rescheduled to today due to our speaker's son's medical emergency on the original webinar day, and I called it on UCLA day. Life is full of ups and downs, as well as unexpected surprises, disasters, good lucks, and discovery. The important thing is to accept such event. I also heard today another speaker has flooding event, unfortunately, this week in her lab. And, that is, I kind of amazed by or shocked by, we are living in a developed country, but we still have that kind of, silly or, unfortunate, things happening to us. But you know what, important thing again is to accept such events and then move on positively, because we we can have or seek the second or third chances. Unpredictability is a pain in life and research, but life may be more enjoyable due to it, or unexpected thing probably lead to a huge important discovery, I believe. Otherwise, or if everything is predictable, including our death and destiny, there would be no meaning of life or hope to improve or live a better life. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Elisa Franco. She is a professor at UCLA, well known by her work on phase-separated RNA organelles. She kindly invited me to give a seminar last year at UCLA, allowing me to meet many SynBYSS speakers, including Professor Paul Weiss, Yi Tang, and Junyoung Park, and some other people. So Elisa, thank you so much for your great contribution to the education and research community, and the virtual podium is all yours now. And thank you again.
All right. Thanks for the invitation and for the introduction. I will complement it by saying buongiorno to anyone Italian watching us. And okay, let me share my screen. Okay. All right. Can you all see my slides?
Yes.
Okay, great. So again, thank you. And today I want to tell you about recent work we've done toward making artificial organelles inside living cells. And, our lab has been working in the space of DNA and RNA nanotechnology for many years. So this field starts from the idea that you can make synthetic DNA. And when you make synthetic DNA, you can decide the sequence of bases arbitrarily. So now you're not tied anymore to what is the context of genes that have a biological meaning. You can actually program sequences as you wish. And you can take advantage of the well-known base pairing properties in DNA strands to build a variety of artificial materials. So people have built machines. So this cartoon represents the fact that if you design correctly a bunch of short DNA strands, you can actually get them to undergo conformational changes over time. And you can design something that looks like a tweezer, and you can open it and close it. And this is a very good example of a nanoscale machine. Now, you can also take advantage of this idea of base pairing to make things that self-assemble. So if you make a four-way junction with four separate DNA strands, that have sticky ends, you can make them assemble into a lattice. And through this very ingenious idea, you can scale up methods to build structures up to megadalton in size. So here are a few examples of how you make, for example, DNA origami. You fold a really long DNA strands with short staples, and then you can form things that look like from a smiley face to a strut, and things that can be really huge. Now, these structures are really amazing in the kind of nanoscale precision and also in terms of size. But they all have one small issue, that is, you always need to anneal them, because as you can see, the complexity of these things requires you to heat up the sample to break any undesired bonds and then cool it down slowly to reach that minimum folding energy that they will have when they achieve the desired complementarity. So now this creates a bit of a roadblock to, how do we use this technology in living cells? Because we cannot actually make something so big and so complex fold at room temperature inside the living cell. So the question that has been motivating me for many years was, well, nucleic acids nanotechnology gives us so many great ideas, but can it help us build structural components in living cells? And so this brings us to, what we have been doing in the last few years. We came up with a very simple, basic structural shape that we call an RNA nanostar. It is basically a single strand of RNA that has, it's a sequence of stem loops. And the stem loops interact through their, loops that, we all know about the HIV kissing loop, but those loops can actually be arbitrary complementary sequences. And the nice thing about these nanostars is that they fold as they are being transcribed, because those stems will form, they're proximal. The stem sequences are proximal, so they will fold immediately as the polymerase is going through the DNA. And now, if you design these kissing loops correctly, you can get these nanostars to form what we call condensates. So these are drops of RNA that are dense in RNA content, you can make them grow at room temperature. And so we have recently published a paper on this. The leading people were Shi Li and Anli Tang in our group. But I should say that, as sometimes happens in science, multiple people have the same idea across the world. And so randomly, we discovered that the group of Lorenzo Di Michele at Imperial College, now at Cambridge, they came up with the same idea independently from us. And then we actually started working together. So this kind of fortuitous situation where we both came up with the same idea led to actually two really great publications that explain how we came up with the design and investigate the design parameters, and then how do we use it to form artificial condensate in synthetic cells. So now, why do we care about condensates? That's a big question. What are even condensates? So the field of condensates has had a renaissance since around 2009, when Cliff Brangwyn demonstrated that there are certain organelles in embryos that behave like liquids. So this video here is a C. elegans embryo, and those green dots are P granules. And he demonstrated that they have a liquid behavior that can be described through the physics concept of a condensate. And then after this study, there's a huge follow-up of people studying what kinds of condensates emerge in a cell. We have this view of a cell being some cytoplasm where things are well mixed, but in reality it's not. There's lots of aggregates of molecules. One of them is, for example, the nucleolus. The nucleolus is a condensate that has even its own structure, where you can have specific proteins enriched in specific phases of that liquid drop. And it's thought that each of them actually organizes specific reactions. It's sort of like an organized microreactor. And there are so many condensates, and each have distinct functions and also composition. The function depends on the composition, which typically includes multiple RNA species, proteins, and small molecules. So, okay, so this is why we care about condensates, because we're just beginning to discover what are their roles in regulating cell fate. And so with our artificial condensates, we use the principles of DNA nanotechnology to design many variants of these nanostars. And so in the paper I mentioned earlier, we characterize, what happens when you change the kissing loop variants, when you change the, different protocols of assembly and the arm length and so on. And we also created a series of orthogonal variants. So you can get condensates that essentially are structurally identical. The motifs are structurally identical, but because they have distinct kissing loops, now they will form condensates that do not mix, and they recruit molecules that then fluoresce in different spectra. But this is kind of like what we've done in the past. Now we wanted to think about whether these condensates could form in living cells. And so we figured out, a way to embed these condensates into plasmids and then transfect them in mammalian cells. And here is example images of what the cells would look like if there was no plasmid. In the middle, it's a cell that expresses with the same plasmid, but you only have a fluorogenic aptamer encoded. So you see that it's diffuse fluorescence. But then when you use the nanostar motif together with the broccoli aptamer, the fluorogenic aptamer, then you can see you have the formation of these big green spheres that are our condensates. And so, now taking advantage of these designs that we have with many orthogonal nanostars and even nanostars that partially interact, we were able to make multi-phase condensates in living cells. So this is where we begin to see how structural DNA nanotechnology can help us. And so here, we were dosing basically the number of red and yellow nanostars, and with linkers that would kind of tell us how in proximity they should be. And then I'm going to show you a movie that gives you a sense of how these structures are really three-dimensional, and they're found primarily in the nucleus of the cell for this specific design. So I just want to wrap up saying that, we're really excited about this prospect of using RNA nanotechnology to control the fate of living cells. And so we have a way to control the formation of these condensates, what proteins they can recruit. We can make multiple orthogonal nanostars and also create subpatterns. And so we would like to turn these into artificial organelles that can localize reactions and program functions, but also use them for biosensing because they're so nicely visible. Perhaps they can help tell us something about the cellular state. And that's it. So I want to thank everyone in our group who has been working so hard on this project, collaborators at UCLA and Cambridge, and the funding agencies that allow us to pay for everything we do. And that's it.
Thank you so much. This is amazing. So I see a lot of potential future application out of this, amazing technology. So thank you so much for amazing talk.
Yeah.
All right. So now the main speaker of today with a longer introduction. Dr. Justin English is an assistant professor of biochemistry at the University of Utah, where he develops and applies new methods in molecular engineering to study G protein couple receptor pharmacology. Dr. English received his PhD at the University of North Carolina at Chapel Hill in the Department of Pharmacology with Henrik Dorman. He remained there for his postdoc in the same department with Dr. Brian Roth. He's an NIH DP2 New Innovator, the most competitive and prestigious award from NIH for junior faculty, and also Eli Lilly LRAP awardee. I met him at the Synthetic Biology Gordon Research Conference, and we had very interesting and productive discussions there. And after that, I recruited him to give a talk for the SynBYSS seminar because I was so impressed by his, depth of knowledge and also broad, kind of, interacting, broad kind of knowledge, he has. So Justin, thanks so much for our exciting discussion last year and your time today. Please take it away, and thank you again.
Yeah, and thank you for having me. This is really exciting, and I apologize that I missed our last session, but thank you for everybody who's going to watch this talk and reach out to me if you have questions and things that you're interested in knowing about from our work. So I guess I'll be talking about several new approaches to accelerate pharmacological discovery and things I've been working on both in our lab and with collaborators. I need to start off by putting on financial disclosures. I own my own company called Evolution Bio, and I also work directly with Eli Lilly. None of the work that I'll be talking about today is directly related to those projects, but I want to make sure that I put that up front. So first, the folks that are really making the work possible. These are my team members, as well as my collaborators and my funding. I work with a large group of people from all over the country on different applications and biological processes. And that's mostly because the way that I see biology is sort of a bit of a mess. It's a tangled series of all sorts of questions that individual people are looking at and trying to solve in really discrete and really focused ways. And there are a lot of really talented folks out there that have very directed and sort of focused applications or questions that they're asking. And a lot of the time when I'm thinking about what kind of projects to pursue, I ask, what is the question that everybody is asking in different ways for the same topic, and how do we design tools and systems that could approach those problems in kind of universal ways to solve many different challenges people are facing? And we do that through basically two pretty simple thought processes or exercises when we approach a project. How do we identify the inputs to a biological system and measure the outputs? And how do we do it in a way where we can effectively, broadly apply that to a lot of different questions that people are asking? I tend to focus on pharmacological systems. I think it's absolutely intriguing how a cell can take a binding event and turn it into biochemical information that gets integrated into the cell in a way where there's actionable change, that we can engineer small molecules that engage with specific targets in the cell and then change the overall behavior of that cell to remedy a disease or to improve its relative health or function is something I've always been very fascinated by. It's why I got my PhD in pharmacology. I'm in a biochemistry department, but I can't honestly say that I've purified more than one protein in my entire life. So I'm a biochemist, but I'm a pharmacologist. And so we think a lot about these processes. And so one of the kind of universal questions in pharmacology is, what are ligands binding to? And how does that binding influence activity? So where are they binding and to what? And what has often been an individual target, but actually any drug-bound target has to interact with other things to impart efficacy. It's not in isolation. And you know, there are some great examples like aurora kinase and its integrated activity in cellular division, or trametinib being a glue for KSLR1 and MEK, which was developed and discovered after trametinib was actually approved as a molecule, and it has these molecular glue-like properties, or JQ1 binding to BRD2 in the Bro domains, and it has these very drug-accessible complexes that change upon drug binding. And so kind of this broader question in pharmacology has always been, what are the drug-bound complexes of a drug target? And so with Fleur Ferguson at UCSD, she's done incredible work with degrader chemical biology and medicinal chemistry over her career. She had these really great bifunctional ligands that could directly bring one protein, usually a degrader or some other ubiquitin ligase, to a target. And I think she and I were both on the job market at the same time. We were both giving interviews, dueling banjos at I think we were at Columbia at the time, and she showed the work she was doing, and I thought this would be awesome if we could just get like proximity labeling instead of degradation. Maybe we could start to map out what the drug-bound complexes in cells were. And once she and I both started our labs, we started this project trying to engineer a system that would allow us to basically translocate a small molecule to its target and biotinylate all of the associated proteins in the drug-bound complex and pull them down and identify them. And so we were successful in doing that. This is the paper at the bottom, and this is just one example. So I need to get my pointer out. There we go. So we have this ortho AP1867 that binds to this FKBP12 F36V mutant. And that's bound to a biotin ligase that is now biotinylating any protein that it's near. And we're bringing it in proximity to a drug target so that we can biotinylate it and then enrich through proteomics and see what the directly bound drug target is, but also its associated protein partners. So we have a linker and then some compound of interest. This is just an example, JQ1, which binds to BRD2, 3, and 4. And we show that when we pull down, we can pull down those primary targets, but we also pull down many proteins that are known to be associated with the BRD2, 3, and 4 regulatory complex in our pull down. And there's just a Western blot of this kind of mass spectrometry data showing that we can, we get some background label, but then we can get very selective label when we add our compound that's the bifunctional. But then we can also do cold competition. So you can add the intended drug back, block up the sites, and then reduce your labeling of that particular target. And so all of our studies are done both with cold competition and enrichment of our individual targets. And this is kind of how we move forward with identifying drug-bound protein complexes with these molecules. We're going in a pretty exciting direction with this, which is to use this to begin to map out how individual small molecules influence drug-bound protein complexes. This is just one example where the MEK inhibitor, cobimetinib, for the most part, only engages with MEK, and then there's a small number of proteins that are associated with MEK. But then there are these molecules called molecular glues that are basically bringing MEK together with KSR1, and that's actually changing the overall drug-bound complex. So while it's a glue that's bringing in MEK and KSR1 together, we're actually identifying additional proteins in these bound complexes that are unique to the glue-based forms rather than the regular MEK inhibitors. And so one thing we would really like to be able to do is make animals that contain this system and be able to influence the activation of the biotinylation in any given tissue. So you can take a small molecule and you know it has an off-target or you know it has efficacy, but you're not sure exactly what the target or the complex might be where the efficacy would exist. You can actually label this and you would give it to an animal, and then you would be able to identify in any given tissue what the drug-bound complexes are. And so Alice Ting was very generous to give us some new versions of her optogenetic turbo ID that she has. And actually, I've mislabeled this. It says mini turbo, but it's actually in the full length turbo ID, where she has a love domain, and she's published this recently, but now she also has a nanoluck fused to that as well. And when you add a substrate that activates the nanoluck, you can optogenetically activate your love domain, which activates the mini turbo, and it allows for chemogenetic labeling of any drug-bound complex. And so we've used this and made a large number of complexes, some that are not light activated that we had previously used, and now ones that are also activated by this substrate with a nanoluck. You add biotin, you add light, but you don't add compound 1, and then we probe for BRD4, and we see some background labeling, and then we apply our compound, and we get much greater enrichment of our target. And so we can now use a chemogenetic, optogenetic kind of combination to label these drug-bound complexes, which is going to be essential if we want to do full tissue distribution of this tool. And so then we were able to find that if we just dilute this construct a little bit, because it's so bright, actually, you can see the nanoluciferase coming out of the wells. And so obviously there's too much construct, which is leading to some of this background. If we titrate this down, now we can get very exquisite drug-bound complex labeling of BRD4 in this chemogenetic system. And so now we're developing both Cree-based mouse lines and xenografts to do this labeling in vivo so that we can see what the drug-bound complexes of different molecules are in animal tissues. And so that sort of is one of the first questions that we developed in the lab is, if we can understand where ligands are bound and what they're influencing, can we get a better picture of what's happening cellularly in a pharmacological context? And so then some of the other questions that we really wanted to ask are what happens after these drugs bind to their individual targets, and what is the signaling modalities that are involved? And we've been studying this problem for an extremely long time, both in our lab and in my postdoc, but also historically in the field of pharmacology. The question has always been, what kind of tool can we develop to measure a cellular readout? And so we have a lot of these, both narrow and mechanistic and broad and very comprehensive, to try and catch what's happening cellularly when you apply a small molecule to the cell, and what kind of information and communication is it imparting. There's this great tool that's used pretty broadly right now for G protein coupled receptors called TruePath that I helped to develop. where the receptor binds to a heterotrimeric G protein, and once it's agonist bound, it disassociates the alpha from the beta gamma, and it's a Brett-based biosensor that basically detects when this alpha beta gamma are disassociating. It's a very specific measurement. We're looking at a pair of proteins that are bound, and we know they're a signaling complex, and then they're disassociated and they signal. And we can measure, exquisitely what the dose response of activity is for any given molecule. This is the mu opioid receptor responding to a whole slew of different clinically and endogenous molecules that it is agonized by, and these are actually mutations in different human beings that carry variants, and we're looking at how drugs bind to these receptors and alter their relative efficacies and potencies. This is work done by my graduate student Ping Guo. She's done an excellent job of beginning to profile like this very nuanced question of what happens when a drug binds to a receptor and that receptor has a mutation that alters its overall pharmacology. So that's a very focused question, and we can kind of get at these aspects of specific cellular biochemistry and the pharmacology underlying them. But one thing that we've had actually a pretty difficult time doing is identifying non-canonical systems. You can only measure what you know. So we know the heterotrimeric G protein binds to the receptor, and so we can design a sensor that reads that out. But for anything where we actually don't know the cellular signaling interaction, it becomes extremely difficult to design tools to measure them. And these are becoming a much more important and relevant component of the GPCR pharmacological field. We're identifying non-canonical interactors, signalers that are not G proteins, but are still binding to the underside of the receptor. Ben Myers here at the University of Utah has done great work showing that PKC directly interacts with smoothen at its catalytic subunit and gets sequestered. And these are all things that are not canonical, that we don't really understand, but have been really hard to discover in sort of a piecemeal fashion. And so I think traditionally people use RNA sequencing as a broad catch-all, right? You want to stimulate, look at what's happening in the genome and read that out. I think one of the difficulties with that is that transcription factors are talking to individual genes together in concert. The cellular signaling pathways that are acting them are then interfacing in an environment that has a lot of post-translational modification, a lot of editing and episomal changes to the overall genome, and epigenetic regulation, distal regulatory elements that are binding to the transcription factor sites. And so you have a very complex second level of logic after you get activation from upstream signaling pathways. It's very difficult to take transcription factor binding motifs and turn them into synthetic reporters directly out of the genome. Lots of great work has now gone into computationally trying to derive these. One of the things that we've been working with Octant, which is a company out of the Bay Area with Sri Kasuri and Henry Chan, is can we take known high affinity binding sequences of transcription factors and integrate them into synthetic libraries where we can directly read out whether or not a particular transcription factor has been activated, sort of divorced from the genetic and regulatory mechanisms of the genome. And so what we've taken is sequences from Gisèle Telepier's lab where they did huge amounts of HT selects, identifying very specific sequences that bind with very high affinity to transcription factors, and integrating them in a massively parallel reporter assay where we can now begin to read out, if we put these sequences that we know bind to transcription factors on pieces of DNA with minimal promoters, can we activate transcription in cells, and what happens when we do that? So we made these libraries with individual transcription factor binding motifs, essentially, that have a uniform length, one after every basically DNA helix, so that they're anti-parallel to one another with these individual spacers in them, variable distances from variable promoters, and then a barcode after, so that if a transcription factor is activated and it engages with these TREs, it will activate the barcode and we'll be able to read that out in next-gen sequencing. The entire paper is currently on bioRxiv, and it'll be published in Nature Communications shortly, I hope, within like the next couple of months. So if anybody's interested, they can read this paper to see more of the details about how we engineered this system. But we got about 6,000 synthetic promoters with 3.6 million barcoded constructs. So we have a lot of redundancy in the system, which allows us to get very refined understanding of what's happening in activation of these individual constructs. And so we take our library of all these barcoded constructs and we transfect them into cells. We provide any type of stimulus, and then we can harvest and deep sequence the barcodes off of this information. So this is just a classic example. Forskolin activates adenyl cyclases. Those adenyl cyclases create cyclic AMP, and the production of that second messenger in the cell leads to activation of various transcription factors, including MAFB and CRE, the cyclic AMP responsive element. And we see these are being enriched very robustly in our library. There's also a large number of other transcription factor binding motifs that are being engaged and activated with the addition of forskolin. These darker blots are all of our negative controls. We have a whole series of scrambles and other non-functional TREs and spacers and things lacking promoters as a way to dial in whether or not we're having a non-specific activation effect. The interesting thing with the whole system is that we can actually pull out each of the individual barcodes from each individual constructs and look at what the relative intrinsic variability of that particular construct was over multiple measurements, its relative amplitude, its strength. And you can pull these directly out of the library and put them into reporter constructs and turn them directly into transgenic luciferase, GFP, or other types of reporters. So the sequence that is in the library can be directly moved over into a reporter plasmid and then used to read out that particular activity in orthogonal assays. So this is just an example of then taking these min-TK, min-promega, and min-CMV versions of one of these MAFB architectures, and then running each one in a luciferase assay to show now we can dial in the relative amplitude of our response. I think everybody wants very good signal to noise reporters, but I think it is also important to be able to have things that have a desired amplitude, and having something that has very high signal to noise, but is always a maximally high signal, can be deleterious for certain applications where having less input but very controlled amounts of it would be desirable. And so you can actually pick through all the individual architectures and responses for any given stimulus in the system and pull out a promoter that has the desired output that you would like. And so we've done this over large numbers of different types of stimuli. These are different kinds of mitogens, metabolites, and small molecule stressors. This is dexamethasone, which directly activates the androgen receptor. When we add dexamethasone to HEK cells, we see the activation of both androgen receptor 1 and 2 binding motifs. When we take those out, we can make 100-fold signal to noise reporter for the androgen receptor with increasing amounts of dexamethasone for one particular architecture. This is another example of looking at both zinc and cadmium. So zinc is a heavy metal, and so is cadmium, but cadmium leads to unfolded protein response. And so we see a great enrichment in heat shock elements with cadmium over zinc, but both activate the metal response element. And so you can imagine doing this for almost any stimulus that you're interested in, to directly take the stimulus type and generate a reporter that then you can use for any other subsequent assays that you might want to know. What we're really trying to capture here is what happens when you stimulate a cell with any kind of pharmacophore, what readouts directly at the transcription factor level are obtained, and how can we use that information to then make reporters where we can do larger scale screenings. We've run this on numerous G protein coupled receptors, as I mentioned, one of our more favorite systems for doing pharmacology. We get this diverse panel of different activations of different transcriptional response elements across a series of G protein coupled receptors and their ligands. One of the more exciting ones that we found was GPR91. This is the natural succinate receptor. GPR91 hasn't had a lot of really great transcriptional response elements. It is thought to be GQ and GS coupled, and there's a good cyclic AMP readout, but it's been known for a while that it has the kind of diversity and divergence from GS-related signaling that hasn't been easy to pin down. This AP1 reporter has been previously used, the commercial one, and it was thought to be a pretty significant unique readout for GPR91. But the signal is actually quite poor. And so we were able to actually engineer an AP1 transcriptional response element for GPR91 that's really well coupled. And then we were able to do a lot of additional studies with GPR91. I won't go into those in significant detail, but what we found is that for any given receptor, whether they have coupling that is pretty similar to other receptors previously thought canonically or not, we're getting these very unique signatures across all different G protein coupled receptors with unique transcriptional response elements associating with individual receptor types. So while they classically might be GQ, GS, GI, even among their same families. When we analyze them in this platform, we're able to dissect out unique signaling properties of each one. And so now we're chasing these down, trying to understand where these come from. All of our data is currently online. All the plasmids to run this system are on Agene and can be ordered directly. All the code for analyzing the data is also available in a Dockerized container on our GitHub. So if you want to look at any of this, please feel free. On this visualizer, you can actually choose from any of the publicly available data that we have, and then directly click on it, and you'll be able to, if I can, maybe I can even show you after the talk. You can actually grab the sequences that are associated with these constructs. It'll print out the oligos that you need to order to basically anneal and drop into the reporter constructs we have on Agene, and you can use them directly as reporters for anything you might want to measure. So that technology is really useful. We're able to actually measure and acquire these really high signal to noise and dynamic range reporters. And so we began to collaborate with a number of people on different applications for this. And one that I'm going to be talking about is really great work done with Willow Koremastis at UCSF. So anybody who's not familiar with Willow's work, he's really revolutionizing deep mutational scanning and the ability of coupling deep mutational scanning of target proteins with their pharmacology and their function. And it's really exciting work. We were fortunate enough to be able to provide them with some transcriptional response element promoters from our TRE-EMPRA that had a large enough dynamic range that they were able to actually pharmacologically discern different small molecules in their flow cytometry assays. So what he tends to do is take a G protein couple receptor, activate it, and let's say it turns on cyclic AMP to activate the Krebs transcription factor. It produces this EGFP with a degron that they've engineered, and that EGFP is then degraded but can be stopped with TMP to stop the degradation of that fluorophore. And then they use that in flow cytometry to identify individual cells with specific pharmacological responses. Using that transcription reporter that we provided with them, they were actually able to differentiate the pharmacology of different ligands for the beta-2 adrenergic receptor. So ICI is an inverse agonist that should impair the ability of beta-2 adrenergic receptor to be activated. DMSO is the vehicle, and then alpranolol, BI, and forskolin. Forskolin is the positive controls activating cyclic AMP in the absence of the receptor. We're actually able to select on a population that now is becoming activated when agonized, which wasn't previously possible in a lot of the paradigms that have been used, even for classic pre-reporters. They then leverage this to look at this really interesting receptor, GPR68, which is a pH-sensitive G protein coupled receptor. It is activated in acidic conditions. So if you move it from pH 6.5 to pH 5.5, it's actually activated, and you get cellular signaling and the production of cyclic AMP. He was able to show using these reporters that they were able to enrich on an active population. So what they end up doing with that, and they can get really discrete dose responses for individual cells within flow cytometry, is do deep mutational scanning using this readout, right? So they make 26 mutations per every position and every amino acid in the G protein coupled receptor, substituting one amino acid for all other possible amino acids simultaneously into this pool of over roughly 10,000 design mutations that they put in this clonal landing pad. Then they take their pooled library and then they screen it using agonists and adding TMP here to group it into bins and look for gain of function, loss of function, or wild type responding receptors. From that data, they were able to find a fitness landscape for the entire receptor, and that data is now here. So now for every individual mutation that you could possibly make, you have a functional readout. And that functional readout is coupled to the transcription reporters that we're generating for individual G protein coupled receptors. The data and how to visualize and look at it is here on our GitHub as well. So you can navigate this data, look at its consequences, compare it to different effects on surface expression, folding, and relative activity with agonism. This paper will be out soon, but it's on bioRxiv if anyone is interested in it or the methods. And Willow has these really great papers on his DIMPLE method for making these deep mutational scanning libraries. The reason that I bring this up is because now what we're really trying to do is figure out how we can improve this particular part of the functional deep mutation scanning workflow. Right now, it's through flow cytometry. So we're separating the cells into like these aggregate bins and saying, okay, this one was relatively more active than the other. But what our lab is engineering currently is how do we turn this into a continuous multiplex next generation based readout of pharmacological activity for every mutation in this library. And so what we actually need are a panel of different transcription factors so that we can read out in a complete fingerprint of signaling activity for an individual molecule where we're getting maybe 5, 6, 7 different readouts simultaneously, each one providing some level of amplitude barcode. And then how do we take those barcodes and append them directly to the mutant that's driving them? And so this has been something that we've been trying to do over the last year. And we've tried a bunch of different methods and ways of concatenarizing these together and doing transplicing. But actually, this really great paper came out from Arvind Asokan recently that we're now collaborating with them on, where he's made this excellent system. If anybody's working in RNA and wants to splice RNA in controlled ways, I'd really recommend this CRISPR-Cas13 system called CRAFT. So Arvind has made a system that allows you to essentially append a new RNA molecule in place of an old one to junction new molecules together. And so we're working on this junctioning approach to try and multiplex deep mutational scanning off of multiple different transcriptional response elements from our TRE-Empress system into a discernible quantitative analysis of how much output from any given TRE we're getting for an individual cell, so that we can kind of take the temperature of cellular signaling for any individual cell for whatever kind of mutation or treatment or molecule that cell might contain. And I hope these multiplex libraries will be of significant use when we finish them. If anybody has suggestions, I'd really like if people reach out if they have other ideas of ways that we can multiplex these kinds of things together. I think it all needs to be an RNA. We need to be able to directly sequence that RNA, and then we need to be able to identify both what mutation or what identity of the cell we have paired with the relative quantitative readout of these different sensors. And so that's all I have today. It's a 30-minute presentation. If people are interested in some of the work that we're doing, we're always hiring and looking for people excited for working in the interface of synthetic biology and pharmacology. I'd like to thank again my team members, collaborators, and funding, and I'm happy to field questions.
Fantastic talk. This is the very important and also exciting, area of research. I want to ask Elisa if she's still there to ask or comment.
To ask a comment on what? The research in general?
No, anything, ask question or comment if you have.
I don't. This is pretty far from my expertise, but it's amazing that you can get this level of control over the receptors. So
Sure.
yeah.
So in that case, let me ask the question. So I also, this is a little bit different from what I'm doing, but I'm generally interested in those kind of signaling process and etc. And but still I want to know to go to, let's say, your holy grail in this field. I believe there are many challenges associated to be there. Also, at the same time, there are a lot of opportunity, when we talking about the Holy Grail. So could you kind of elaborate a little bit more, what kind of more challenges we should overcome to be there, and then what kind of opportunity we could have, I see some, Shuri Kosuri, I mean, a good friend of mine, and then working with you, I guess. But I know, he kind of tried to solve some problem in the synthetic biology field, but I just wanted to hear from you.
Yeah, I think the holy grail
Yes.
of probably pharmacology generally is, can we computationally derive molecules with the desired molecular efficacy directly without needing to experimentally define each one? I mean, I think that's where everyone is trying to go.
Mm-hmm.
I think a lot of the docking right now is based on static structures. that don't contain a dynamic or allosteric element. They're, for the most part, looking at something that and it's a huge revolution relative to where we were even 20 years ago, right? Our ability to now computationally dock large molecular virtual libraries of molecules into a target, and then even come up with things that have affinity is a major advance, right? But I think our current challenge is that while we can make molecules with affinity, we cannot really rationally design molecules that have the desired efficacy that we would like. And so, for example, what I was talking about with how these G protein couple receptors can signal through many different pathways. They can signal through different G alphas, but they also have many other effectors. And what effector they communicate with, so a classic example is the mu-opioid receptor. Morphine does great in analgesia, but also leads to respiratory depression and death in people who take an excess amount. It would be phenomenal to be able to avoid the respiratory depression, but maintain analgesia, right? This has been something that people have been attempting for the better part of 15 years since this was discovered and really kind of mechanistically understood. But it's very difficult to design molecules that have that level of rational control over one pathway or the other. And I think what we really need to begin to do is fill in how structure and state of a receptor conveys information that is unique. And once we have that information, we can begin to more rationally design molecules that stabilize states that convey that information. And it's the same for G protein couple receptors as almost any other drug target you can imagine. But it's a pretty big lift. I think if someone could come up with a way to reasonably apply and understand molecular efficacy and allosteric logic to every protein target that a molecule binds to, we'd be in pretty good shape.
That's thank you so much for that insightful answer. Actually, my lab also tried to use G coupled receptor in yeast, and unfortunately the postdoc left without finishing up. And then, but she a little bit struggled because this is not easy protein to work with. That's how that's my impression based on my conversation with her. But the one thing I a little bit optimistic is, now everyone talking about AI, and we have AI Nobel Prize winner this year. Actually, multiple. And then given that, promising, achievement, what would be your kind of prediction regarding our fear in the future? Your fear in the future, using AI, whatever, prediction algorithm, structure, prediction algorithm, etc.
Yeah, I think we're getting there. Brian Roth and Brian Choquette put out this beautiful paper where they took the structure of a receptor, or the alpha folded version of the structure of that receptor, and docked to it, and were able to get molecules of equal potency and relative activity against the folded computational model as the resolved one. But it really depends on the target. The same can't be said of doing it for a receptor for which there is no folding data. Or structure. Is much harder. People are attempting it for different computationally derived models. A good example is MRGPRX2 and MRGPRX4, which they also solved, where prior to solving it, it was very difficult to molecularly dock to a computational model, but after solving it and refining their model, it's dockable. And so understanding kind of what we're missing in that space right now between the folded and then the functionally relevant active kind of dynamic state of the receptor is probably the big next leap in the computational space for AI-induced drug binding. And I think we're going to get there.
That means the drug company or big pharma still rely on high throughput screening.
Yeah, I think I think that they can get new candidate molecules against a particular target with their binding, but we still are not able to then say that molecule is going to have the desired efficacy, only that it will bind, right?
I see.
The rational design to say that it's going to bind and it's going to be a partial agonist or an allosteric modulator in a positive or negative direction is very is still out of reach for these computational approaches.
You know what? I have some trauma in terms of protein engineering because when I was graduate student, I literally actually we rely on very high level molecular dynamic simulation, the basically atom level simulation, and then we design some protein variant to change the specificity of the enzyme. And then, but still that, combination gave me 20,000 different variant. And we synthesized all of that using, at the time, we didn't call Golden Gate, but still Golden Gate method, sense. And then, making the library of 20,000 targeted variant, but I need to pick 40,000 colonies by hand with colony picker. And then I become robot to screen all of that by multi-channel, pipette without, again, the liquid handler. And then after that, many months of the screening, I realized, okay, I'm not gonna do protein engineering ever. And then now we have AlphaFold and also Rosetta, and more reliable. And then I also bought a liquid handler, and then I gave that on to my student, and basically we start a protein engineering, project, many years later. And then I was so amazed by the power of the, those algorithms, but still, we are not there yet, you are saying.
Yeah, I think for enzymes, definitely. I think for enzymology, they're engineering some really fantastic molecules. I saw a great talk by one of the folks that's in one of the spin-off companies from David's group recently at St. Jude, and it was,
I believe I probably watched the same thing because I attended CRISPR conference this week.
Yeah, okay. With the VX gas and like degrading VX and everything.
One of them is the St. Jude, in the St. Jude hospital.
Oh, okay, yeah, different group. Yeah, it's I mean, they've done phenomenal work with some basically rational enzyme engineering. I think we just need to get more of the rules of the game of how structural biology and molecular dynamics for GPCRs convey information to get it to that level for enzymology of some of these other targets that are being designed.
Also, my work is about sensor development using transcription factor. So a bit different one, a bit probably, different difficulty than the enzyme engineering. But now I'm doing more protein engineering because of that, liquid handler and the protein design tools available in the, in here.
Yeah.
Yeah. Okay, so Elisa, do you have any, additional comment, question, anything? Otherwise, I will close.
Oh, that's it. I want to say AI is not as big yet in the DNA nanotech community. I think our mantra is more like rational design. But that said, predicting RNA tertiary structure is pretty hard. So I can see how AI it will help a lot in that sense.
So you use mostly UPEG or kind of program, right?
Yeah.
Yeah, we also done around, for more than 10 years RNA. That's the only thing. And then also you could use, I mean, the sequencing, the Julius Lux, and so those people kind of develop, after cross-linking, do the sequencing. In that way, you could predict or estimate the tertiary structure or whatever, right? Shapeshift. Thank you. Yeah, shapeshift. That's what the word. Anyway, so still that's the future thing we wanted to see. But I hope, I mean, somebody can improve the prediction ability more. In that way, we not feeling like doing art, but more engineering. That's my dream. Sometimes I feel like I'm an artist. So, okay, so let me close. Thank you all for joining and staying today. We'll meet again on October 24, the normal time, the Thursday, the same time, the same Zoom link. We'll have Professor Seth Lakoff Nahom at Boston Children's Hospital and Harvard Medical School, and Dr. Jae-sung Cho at MIT. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with us. And thanks. And I stop recording. 1 second. I don't know. I suddenly disappear, right?
You did.
Oh, I don't know. So I just stopped recording. My computer now act weird.
I got a message that actually you're still recording, but
Yeah,
No.
I went back on again, I think.